Hello everyone and welcome to today's session. And today we'll be discussing the NCERT Exemplar questions for the chapter Human Health and Diseases. So this is the part 1 of the lecture. This is the disclaimer. Okay, so let's understand what are the objectives of today's session. So today we will understand the basic concepts related to the chapter Human Health and Diseases by working through NCERT Exemplar questions. So these questions, they are based on some concepts. We'll discuss these concepts by discussing the NCERT Exemplar questions. Okay, so these questions have been categorized into these categories. So we have multiple choice questions, very short answer type questions, short answer type question, and long answer type questions. Okay, so first question is, the term health is defined in many ways. The most accurate definition of the health would be. So they are asking the definition of health here. Okay, so you have 4 options, A, B, C, D. You have to select the correct answer from these options. Now the first option is health is the state of body and mind in a balanced condition. Now second option is health is the reflection of smiling face. Next question is health is a state of complete physical, mental and social well-being. And the last option is health is the symbol of economic prosperity. What will be the correct answer? Let's have a look at the answer. Option number C is the correct answer. Health is the state of complete physical, mental and social well-being. So, as we have discussed, health is the state of complete physical, mental and social well-being. Now, not merely the absence of disease or infirmity is the definition of health from the World Health Organization. Health is more than just the absence of disease or physical fitness. It is a state of complete physical, mental and social well-being as defined by World Health Organization. Okay, so basically health is the overall well-being. Now the definition of the state of complete physical, mental and social well-being is widely attributed to the World Health Organization. So this definition states that health is more than just the absence of disease. It encompasses a comprehensive state of well-being across physical, mental and social well-being. Okay. Now, when people are healthy, they often experience improved mood, which can lead to a more positive outlook and increased confidence. Now, this increases the productivity and brings economic prosperity. It also increases longevity of people and reduces infant and maternal mortality. Okay, so here you can see the mortality means the death rate is being decreased here. Both infant and maternal death rate, it is reduced. Next question, diseases are broadly grouped into infectious and non-infectious diseases. In the list given below, identify the infectious diseases. Okay. So, first one is cancer, second is influenza, third is allergy, fourth is smallpox. So, the options are given to you. You have to choose the correct answer from these options. The first option is A and 1 and 2. Second option is 2 and 3. Third is third and fourth. And the fourth option is second and fourth. What will be the correct answer? So let's have a look at the answer. Now, influenza, commonly known as flu, is the infectious disease of birds and mammals caused by influenza virus. The most common symptoms are chill, fever, running nose, sore throat, muscle pain, headache, coughing, weakness or fatigue, and discomfort. Now, these symptoms are your body's way of fighting off the virus. Now, cancer is defined as an uncontrolled division or proliferation of the cells without any differentiation. This means the cells keep multiplying without becoming specialized, which is what makes cancer dangerous. Okay, so we have talked about influenza and cancer now. Okay, so cancer, it is a non-infectious disease caused by agents called carcinogens. So carcinogens, they trigger cancer. Now let's talk about the smallpox. Smallpox is serious. It is highly contagious and often life-threatening disease characterized by a rash and blisters on the face, arms, and legs. The word contagious means it is infectious. Now, it is caused by the variola virus. This means smallpox is an infectious disease as it is directly linked to the specific virus. Now, smallpox gets transmitted from person to person through various means such as sneezing, saliva, and contaminated body fluids. Now, let's talk about allergy. Allergy is the exaggerated response, means heightened response of the immune system to certain antigens in the environment like pollen, dust, mites, molds, cloth fibers, and animals' hair. Now, this exaggerated response occurs due to the release of chemicals like histamine and serotonin from mast cell. Okay, mast cell act like alarm systems in the body, releasing these chemicals when they detect an allergen. Now, allergy is a non-infectious response. This means it cannot be transmitted from one person to another. Now, next question. The sporozoites that cause infection when a female Anopheles mosquito bites a human being are formed in. So the options are a liver of human. Second option is RBCs of mosquito. Third is salivary glands of mosquito. And fourth is intestine of human. Okay, option number C is the correct answer is salivary glands of mosquitoes. Okay, so gametocide, they enter in the female Anopheles mosquito when they bite an infected person. Yeah, so gametocide, it is the sexual stage of this pathogen plasmodium. Okay, now these gametocytes, they fertilize in the stomach's gut. Okay, in the insect's gut. Clear? And after fertilization, sporozoites are formed, and these sporozoites then migrate to the salivary glands of the female Anopheles mosquito. Okay, then when again the female Anopheles mosquito bites a healthy person, it will, you can say, transfer these sporozoites into that healthy person. After entering the human body, the sporozoites reach the liver cells where they multiply by sexual reproduction. This is followed by their attack on red blood cells, resulting in the rupture of RBCs. Now, the ruptured RBCs release a toxin called hemozoin, which is responsible for high recurring fever, chills, and shivering. Okay, next question. Many disease can be diagnosed by observing the symptoms in the patient. Which group of symptoms are indicative of pneumonia? So the options are difficulty in respiration, fever, chills, cough, and headache. Option number B, constipation, abdominal pain, cramps, and blood clot. Option number C, nasal congestion and discharge, cough, sore throat, and headache. And option number D, high fever, weakness, stomach pain, loss of appetite, and constipation. What will be the correct answer? Let's have a look at the answer. Option number B is the correct answer. Difficulty in respiration, fever, chills, cough, and headache. Okay, so now let's have a look at the thinking process. Now, pneumonia is caused by a pathogenic bacterium, Streptococcus pneumonia and Haemophilus influenza. Now, these bacteria spread through droplets in the air, especially when someone sneezes or coughs. Okay, so keeping this in mind, you have to answer this question. So let's have a look at the answer. Now, what are the symptoms? Okay, difficulty breathing, okay, fever, chills, cough, and headache. These are the symptoms of pneumonia. And then constipation, abdominal pain, cramps, and blood clot. These are the symptoms of amoebiasis, also known as amoebic dysentery, caused by Entamoeba histolytica. Next, nasal congestion and discharge, cough, sore throat, and headache. These are the symptoms of common cold, also known as influenza. Then high fever, weakness, stomach pain, loss of appetite, and constipation. These are the symptoms of typhoid, which is caused by a bacteria, Salmonella typhi, which infects the small intestine. Next question, the genes causing cancer are, the options are structural genes, expressor genes, oncogenes, and regulatory genes. What will be the correct answer? Okay, option number C, oncogenes is the correct answer. The normal cells have genes called cellular oncogenes or proto-oncogenes, which are present in an inactivated state. Now, upon certain conditions like mutation, these genes can be transformed into cancer-causing oncogenes. Now, structural genes, expressor genes and regulatory genes are responsible for the regulation of gene expression. So they help control when how much of a gene is turned on or off, but they are not directly responsible for causing cancer. Next question. In malignant tumors, the cell proliferate, grow rapidly, and move to other parts of the body to form new tumors. Now, this stage of disease is called metagenesis, metastasis, teratogenesis, and mitosis. What will be the correct answer? The correct answer is option number B, metastasis. So, cancer is defined as an uncontrolled division or proliferation of cells without any differentiation. Now, the repeated division of these cells forms a large mass of tissue called tumors. The tumors are of two types, benign, which are non-cancerous, and malignant, which are cancerous. Benign tumors stay in one place and usually don't cause much harm, while the malignant tumors can invade other tissues and are much more dangerous. The invasion of cancerous cells from one part of the body to another is known as metastasis. So this is why malignant tumors are so serious. They're quite dangerous. Now, what is tetragenesis? Tetragenesis. Okay, this refers to prenatal toxicity, which means structural or functional defects in a developing embryo or fetus, and it is not related to cancer. Now, mitosis, it is a normal type of cell division that results in two daughter cells. In cancer, this process goes out of control. But in healthy tissues, mitosis is how our bodies grow and repair themselves. Next question, which of the following are the reason for rheumatoid arthritis? You have to choose the correct option. Okay, so first one is lymphocyte become more active. Second is body attacks cells. Next is more antibodies are produced in the body. And the fourth one, the ability to differentiate pathogens or foreign molecules from cells is lost. So we have four options. You have to select the correct answer from these four options. Okay, so option number B, second and fourth is the correct answer. Now, rheumatoid arthritis is an autoimmune disease. So autoimmunity is when the immune system starts rejecting its own cells. Here the immune system is not able to recognize between self and non-self, and it starts targeting self cells. The body loses its ability to differentiate between own cells and the invaders. So this confusion leads the immune system to attack its own tissues, which is what happens in the rheumatoid arthritis. Now this results in damage to the body. Now when a foreign antigen enters the body, lymphocytes become more active and produce more antibodies. So this is a normal response to the infection, but in autoimmune disease, the problem is that the immune system is attacking the wrong cells. They are attacking its own cells. Okay, that's why second and fourth is the correct answer. Second is body attack cells, and the fourth one is the ability to differentiate pathogens or foreign molecules from cells is lost. Next question is, AIDS is caused by HIV. Okay, among the following, which one is not a mode of transmission of HIV? The options are transfusion of contaminated blood. Second is sharing the infected needle. Third one is shaking hands with infected person. And fourth is sexual contact with infected persons. So option number C is the correct answer, is shaking hands with infected person. Now let's talk about the transmission of HIV infection. It generally occurs by the first one is sexual contact with the infected person. So this includes unprotected sex, where the virus can pass through body fluids. Second is the transfusion of contaminated blood and blood products. Okay, they can also transmit the HIV virus. This is why the blood banks are carefully screened before the donations. Third one is sharing infected needles, especially among intravenous drug users. Okay, these intravenous drug users, they are very high risk because needles can act like a bridge carrying the virus from one person to another if they are not properly sterilized. Fourth one, infected mother can pass HIV to her child through the placenta during pregnancy. Okay, even childbirth or even breastfeeding as well. So this is known as vertical transmission. Okay. Now, shaking hands with infected person is not a mode of transmission. Now let's talk about very short answer type questions. So here the first question is certain pathogens are tissue or organ specific. You have to justify the statement with suitable example. Okay, so let's discuss the answer for this question. Now, certain pathogens, okay, they are tissue or organ specific because they are adapted to overcome the resistance mechanism of those tissue and organs. Now, let's have a look at the one example. Now, pathogens that enter the gut must survive the low pH of the stomach and resist the digestive enzymes. So this shows how only certain pathogens with the right adaptation can infect the gut while others are destroyed. The next question, the immune system of a person is suppressed. In the ELISA test, it was found positive to a pathogen. Clear? So these are the two conditions that they have given to you. Immune system of a person is suppressed. Okay. Now, the ELISA test was done. In this ELISA test, it was found that the person was positive to a pathogen. So you have to name the disease the patient is suffering from and what is the causative organism of this disease and which cells of the body are affected by this pathogen. Okay, what are pathogens? Organisms that cause diseases. Okay, so let's have a look at the answer. Now, the immune system of the person is suppressed. Okay, this is the first, you can say, symptom. Next, we see that the ELISA test was positive for a pathogen. So, this test is a key diagnostic tool for detecting infections like HIV, which can suppress the immune system. Now, based on these clues, we identify that the patient is suffering from AIDS. So, remember, AIDS is a condition where that results when the immune system is severely weakened by HIV. AIDS is caused by human immunodeficiency virus, okay, which is HIV. Now, this virus specifically targets and destroys important immune cells. HIV is a retrovirus, means it contains RNA as the genetic material. Okay. Now, the main cell affected by HIV are macrophages and T helper cells. These cells are crucial for coordinating the immune response. So, when they are damaged, the whole system is compromised. Yeah, so basically, HIV multiplies in these cells, macrophages and T helper cells, and slowly their number keeps on decreasing. That's why the immune system is compromised. Next question, where are B cells and T cells formed and how do they differ from each other? Let's have a look at the answer. Okay, first have a look at the thinking process. Now, lymphocytes are divided into two main types. T lymphocytes, also known as T cells, and B lymphocytes, also known as the B cells. Okay, now let's have a look at the answer. Now, both B cells and T cells, along with other immune cells, are produced in the bone marrow. Now, let's have a look at the specific differences between B cells and the T cells. Now, B cells, also known as the B lymphocytes, they mature in bone marrow. They produce antibodies and they do not respond to organ transplantation. Here. Now, T lymphocytes, also known as T cells, they mature in the thymus. They directly attack antigens or attach B cells to produce antibodies. Okay. So they promote the antibody production. Okay, by B cells. Clear? And they directly attack the antigens. Clear? Or you can say they trigger B cells to produce antibodies also. And they do respond to the organ transplantation. So basically the organ rejection, it is because of the T cells. So the cell-mediated immunity is responsible for the organ rejection. The cell-mediated immunity involves these T cells. Next question given below are the pairs of pathogens and the disease caused by them. Which out of these is not a matching pair and why? Okay, so you have 4 options. First one is virus and common cold. Second is salmonella and typhoid. Third is microsporium filariasis, and the fourth one is plasmodium malaria. So what will be the correct answer? Okay, so let's have a look at the answer now. So Wicheria bancrofti and Wicheria malayi, they are the filarial worms. Okay. They cause a slow developing chronic inflammation of the organs in which they live for many years. Okay, so these two filarial worms, they are responsible for causing filariasis. So these worms, they live in the lymphatic vessels and they cause swelling of these lymphs, the lymphatic vessels, especially that of the lower limbs. Okay. So here the correct answer will be option number C, microsporium filariasis. This is not a matching pair because filariasis is caused by filarial worms, Wicheria bancrofti and Wicheria malayi. Now, in filariasis, what happens? These worms affect the lymphatic vessels of the lower limbs, leading to a disease called elephantiasis or filariasis. Now, microsporium, this is a fungi. Clear? So, fungi like microsporium, trichophyton, and epidermophyton actually cause a ringworm, not filariasis. Okay. Now, next question is what would happen to immune system if thymus gland is removed from the body of a person? Let's have a look at the answer. Okay, so let's start by outlining the thinking process. Now, remember that thymus is the primary lymphoid organ. Okay. Within thymus, the immune cells develop and mature. Thinking this in mind, you have to answer this question. So let's have a look at the answer. So if the thymus gland is removed, the person's immune system will become weak. This is because the body loses its main site for T cell maturation, which are crucial for immune defense. So within the thymus, T cell maturation occurs, and T cells, they are very important cells of the immune system. So as a result, the person's body becomes more prone to infectious diseases. In this image, you can see the location of the thymus gland in which T cell maturation occurs. Okay, where you can see thymus gland is in between the right lung and the left lung, and it is below thyroid. Next question is, many microbial pathogens enter the gut of humans along with the food. What are the preventive barriers to protect the body from such pathogens, and what type of immunity do you observe in this case? Okay, Now, many microbial pathogens can enter the gut of human along with the food. This is the common way for infections to start, especially if food is contaminated or not handled properly. Now, the preventive barriers to protect body from such pathogens are as follows. So, the first barrier is the mucus coating that lines the gut. Okay, so this mucus, this prevents or it traps the microorganism, preventing them from reaching the cells of our digestive system. In addition to mucus, saliva in the mouth and hydrochloric acid in the stomach play important roles. Saliva contains enzymes that can break down some microorganisms, while the acidic environment of the stomach destroys many pathogens before they can reach the intestine. Now, all these defenses are the part of what we call innate immunity. So this is body's natural inborn way of protecting itself from infection. So innate immunity is from birth. And innate immunity, it doesn't require previous exposure to a specific microorganism. Innate immunity is present from birth and is inherited from our parents. This innate immunity remains with us throughout our life. Next question, what are interferons and how do interferons check infection of new cells? Okay. Now let's have a look at the answer. Okay. Now, in response to viral infections, our body produces glycoproteins called interferons. Okay. Now let's talk about the barrier interferons. So such type of barriers of innate immunity are called cytokine barriers. So interferons come under the cytokine barriers. Okay. Now interferons protect non-infected cells from further viral infections because interferon they are released by virus infected cells. Okay, now we are discussing short answer type question. We have to differentiate between active immunity and passive immunity. Let's have a look at the answer. The difference between active immunity and passive immunity are is the first one, active immunity. It is developed due to contact with pathogens or its antigen. Okay. So passive. What about passive immunity? Passive immunity. This is developed when ready-made antibodies are injected into the host or into the person's body. Okay. Let's talk about active immunity. It has no side effects because body is generating an immune response. But passive immunity may have reactions because you are introducing the antibodies from some other source into the host. Active immunity, this is slow but long lasting because in active immunity, the antibodies are being produced. So it takes time to produce antibodies, but it is long lasting. It has memory cells. Okay. And because of its memory cells, again, there is a heightened response, immune response whenever there is a subsequent infection with the same pathogen. Okay. Now, what about passive immunity? It is fast but lasts only for few days. Now, active immunity takes time to develop its response because antibodies takes time to develop. Okay, passive immunity, it is used when the immune response has to be faster. So in the case of snake bite, these antitoxins are given. These antitoxin consist of these antibodies. Okay, so whenever you require, you can say immediate treatment. Okay, or you require immediate response, then you go for passive immunity. Examples of active immunity are vaccination. Okay, for diseases like polio. In vaccines, attenuated pathogen or antigens of that pathogens are introduced into the host body. Okay, and the antibodies are generated against this antigen or weakened pathogen. Examples of passive immunity is administration of tetanus antitoxins or antitoxins for the snake bite or the transfer of the antibodies from the mother's milk to the child. So these are all examples of the passive immunity. Here you can see in this image the examples of the active and the passive immunity. Okay, so active immunity can be triggered by any infection or by vaccination also. And passive immunity involves transfer of maternal antibodies through colostrum or through placenta. Okay. As well as we can introduce ready-made antibodies into the host. Okay, so monoclonal antibody can be made and it can be introduced into the person's body. Next question is, you have to differentiate between benign tumor and malignant tumor. So let's discuss the answer. What is the difference between benign tumor and malignant tumor? Okay, so benign tumor, it is non-cancerous tumor. Okay, it is not spreading to other regions of the body, whereas malignant tumor, it is a cancerous tumor. Now, benign tumor does not show metastasis and is non-invasive, whereas malignant tumor, it is shows metastasis and thus involves other body. It invades other body parts. Benign tumor, it stops growth after reaching a certain size, whereas malignant tumor, they show indefinite growth. Benign tumor, they show limited adherence. Okay, so in benign tumor, limited adherence occurs among cells of benign tumor. In the case of malignant tumor, there is no adherence among cells. They tend to slip past one another and they keep on spreading. Benign tumor, it is less fatal to the body. Malignant tumor, it is more fatal to the body because it keeps on spreading. It shows metastasis and it can invade other body parts as well and start a new tumor there. Okay, it can form a new tumor there. Here in this image, you can see difference between benign and malignant tumors. Notice how the benign tumor is contained while the malignant tumor invades surrounding tissue. The next question is, prevention is better than cure. You have to comment on this statement. So prevention is always better than cure because some diseases cause extensive damage in the body tissues or organ. Once the damage is done, it can be very hard or even impossible to fully repair. The first point is here, diseases can have a negative effect on the capacity of our organs to function. For example, if the heart is damaged by a disease, it may not pump blood as efficiently, just like a weak battery in a device. Second, some diseases can leave permanent or long-term effects. Okay. Now, this means the person might never fully recover or they may have to live with the limitations for the rest of their life. Third, diseases can also have negative mental and psychological effects. Being sick for a long time can lead to feeling of sadness, anxiety, or even depression. The next one is, there's a lot of financial burden with the person is not able to work and the cost of the medicines, it is very high. Now, so prevention is easier and more effective than curing a disease. By taking steps to avoid getting sick in the first place, we protect our bodies and minds and our wallets. Okay, clear? So this was the answer for this question. The next question, explain any 3 preventive measures to control microbial infections. So let's discuss the answer. Now, preventive measures to control microbial infections include, first one, maintaining personal and public hygiene is essential. This includes following cleanliness standards, proper waste disposal, and regularly cleaning water sources. These actions help remove the environments where microbes can thrive, much like keeping your kitchen clean prevents the pest. Now, second, controlling or eliminating vectors is important. So vectors like mosquitoes and flies can carry microbes from one person to another person. So by preventing water stagnation and using disinfectants or biological control, we can reduce their numbers and stop the spread of disease. Third, proper immunization through vaccination is a powerful tool. Now, vaccines prepare our immune system to fight specific microbes. protecting us and those around us. The next question is maintenance of personal and public hygiene is necessary for prevention and control of many infectious diseases. You have to justify the statement giving suitable examples. Okay, so diseases which are easily transmitted from one person to another person, these are known as infectious diseases. Now, for prevention and control of such diseases, what is required? First of all, education is the key step. So by teaching people about how diseases spread and how to protect themselves, we empower communities to take actions. For example, posters about hand washing in schools help remind students to keep germs away. Second one is isolation. Keeping infected individuals separate from healthy ones it helps stop the disease from spreading. Now, vaccination is a crucial preventive tool. Getting vaccinated helps our bodies recognize and fight off specific diseases before they can cause harm. The next one is sanitation. Okay, so sanitation should be improved to avoid infection from polluted water and contaminated food. So clean water and uncontaminated food are essential for preventing diseases like cholera and typhoid. Improving sanitation is like building a protective wall against harmful microorganisms. Okay. Now let's talk about the eradication of the vectors. So vectors are basically mosquitoes and flies that can spread diseases. So by destroying their breeding grounds and using methods to kill adult vectors, we can stop diseases like malaria and dengue from spreading. Now next one is sterilization. Now sterilizing the patient's surroundings and their personal items help prevent the spread of infections to others. Okay. Now moving on to the next question, the following table shows certain diseases, their causative organism and symptoms. You have to fill in the gaps. Okay. So let's have a look at the answer. Now you can see ascariasis. This is caused by ascaris. And what are the symptoms? Internal bleeding, weakness. Okay, roundworms can also appear in stools and they can be abdominal pain. Next, ringworm. This is caused by trichophyton. What are the symptoms? Appearance of dry, scaly lesions on various parts of the body. Then typhoid is caused by bacteria Salmonella typhi. And symptoms are already given: high fever, weakness, headache, stomach pain, and constipation. Now, pneumonia. This is caused by Streptococcus pneumonia. And the symptoms are given. Symptoms are as fever, chills, cough with mucus and difficulty in breathing. Then common cold, this is caused by rhinovirus, and the symptoms are nasal congestion and discharge, sore throat, cough, and headache. And in the last, filariasis, this is caused by Filariaworm Wucheria bancrofti, and it causes inflammation in the lower limbs because this worm, this affects the lymphatic vessels, especially of the lower limbs. Move on to the next question. Many secondary metabolites of plants have medicinal properties. It is their misuse that creates the problem. You have to justify the statement with an example. Let's have a look at the answers to drugs like barbiturates, amphetamines, benzodiazepines, and LSD are all examples of plant-derived compounds used as medicines. So these are especially important for treating mental health conditions like depression and insomnia. Okay, let's talk about the morphine now. So morphine is a very effective sedative and a painkiller. Morphine is often used for patients after surgery to manage severe pain. Now, let's talk about the misuse of plant metabolites, fruits, and disease. So misuse of plant metabolites, fruits, and seeds in amounts, or if they are frequently used more than the prescribed, for medicinal purposes. This can impair one's physiological or fundamental behavior, creating problems for the society, and slowly moves towards the eternal truth, which is death. Okay, so the misuse of these drugs can affect a person's physical health. Okay, and it can affect the person's mental health. Okay, it can also affect the social well-being. Okay, it can even lead to death. Okay, now next question: why cannabinoids are banned in sports and games? So let's have a look at the answer. Okay, so cannabinoids are banned in sports because athletes might misuse these drugs to enhance their performance. Now, even though some athlete might think these drugs help, cannabinoids can have serious negative effects on their general health as well. Over time, using these substances can disrupt the normal functioning of organ systems. Clear? These cannabinoids are banned in sports and games. Now let's talk about long answer type questions. The first question is represents schematically the life cycle of malarial parasite. Okay, which is Plasmodium, which is a protozoan. So let's have a look at the answer. Okay, let's talk about the life cycle of the malarial parasite. Okay, so this is the image showing you life cycle of the malarial parasite. So we'll start, okay, with the stage when the female Anopheles mosquito bites the human. Okay, so it has two hosts, human host and mosquito host. Okay, mosquito is the female Anopheles mosquito. And this female Anopheles mosquito, this is also a vector for the transmission of this disease. So this mosquito, this is a vector and a host as well. And it has a very complex life cycle because it involves two hosts. Okay, so first is let's talk about when the human is the host. So we are calling it as human stage. Okay, in this asexual reproduction occurs in the human host. Now, these mosquitoes which are infected, whenever they bite the healthy human, they inject the sporozoites into that human. Okay, so sporozoids, these are the infectious stage of this pathogen. Then the sporozoid, they travel through the bloodstream and enter into the liver cells. Now in the liver cells, they will multiply by sexual reproduction. They will form schizons, and from the schizons, merozoites are formed. Okay, now these merozoites, they will enter into the red blood cells. Clear? So these merozoites, they developed in the liver cells, and the liver cell burst and they release the merozoites into the blood, and these merozoites will then infect the red blood cells. Again, they will multiply by asexual reproduction, and then the RBCs will burst. Clear? Now you can see from the merozoites, gametocyte develops within the RBCs only. Okay, so whenever these RBC burst, gametocytes are released. Okay, so merozoites, they infect RBCs, and some of the merozoites, they transform into gametocytes within the RBCs. And when the RBCs burst, these gametocytes are released. Gametocytes, remember, it is the sexual stage of this pathogen. So there'll be a male gametocyte and a female gametocyte. Now, next one is the mosquito stage, where the mosquito is the host. This is a sexual stage because gametocyte, the sexual stage, has been formed. Now, whenever the mosquito, it will bite this infected person, then along with the blood, the gametocyte, they will also be ingested by the mosquito. Now, this gametocyte, they reach the mosquito's gut. Okay, and the male and the female gametocyte, it will fuse together, so fertilization will occur. Okay, after fertilization, what happens? Sporozoites are formed. Clear? So after fertilization, zygote is formed. It develops into oocyte. Oocyte develops into sporozoites, and then these sporozoites, they migrate to the salivary glands of the female Anopheles mosquito. They are stored there, and whenever this female Anopheles mosquito bites a healthy person, this will again transfer this porozoid into that healthy person. So this is the overall life cycle of the Plasmodium. We have two hosts, human host and the second host is the female Anopheles mosquito. And the female Anopheles mosquito, this is also acting as a vector. Next question, what are the methods of cancer detection? Describe the common approaches for treatment of cancer. Okay, so let's have a look at the answer. Now, early detection of cancer is essential because if the cancer is detected early, then it can be completely cured as well. Now, what are the methods of cancer detection and diagnosis? Let's talk about it. And the first method is biopsy and the histopathological studies. So this involves taking a small sample of tissue, blood, or bone marrow and examining it under a microscope. Now, test for increased cell counts. Okay, in the case of leukemia, blood cancer is done. So leukemia is the cancer of the blood. So in this case, what you do? You look at the increased number of the cell counts. So this can show that the person is having leukemia. Okay. Now next one. So doctors also use imaging techniques like radiography. Okay, in which X-rays are used, then CT scan. Okay, and then MRI. Clear? So these techniques are used for the detection of the cancer. Clear? So these techniques are used to detect cancers of the internal organs as well. Now, detection of cancer-specific antigen is another approach. Antigens are like unique flags on cancer cells. Okay, so these antigens are present on the cancer cells. Okay, so these antigens are detected. Okay. Now, molecular biology techniques can also be used to detect genes that make someone more likely to develop certain cancers. Okay. In some individuals, the chances of this cancer is high. Okay, what are the common approaches for treatment of cancer? So there are many strategies which the doctors can use to fight cancer in the patients. First approach is surgical removal of the tumor. So this means physically cutting out the cancerous tissue from the body. Second approach is irradiation of the tissues, also known as radiation therapy. Here, high energy rays are used to kill cancerous cells. Now, the third approach is immunotherapy, which uses substances like interferon to boost the body's immune system to kill the cancer. So we are basically boosting the immune system so it can kill the cancer cells. So what we are using here? We are using a substance such as interferon to boost the body's immune system. Okay, so these were some of the common approaches for treating the cancer. Then chemotherapy can also be given, in which certain drugs are given to kill the cancerous cells. Basically, what is done? A combination of all these techniques, these are used by the doctors to kill the cancer cells. Okay, surgery can be used. In addition, irradiation and immunotherapy can be done. Chemotherapy can also be given to that person. Okay. But you can say it is taken into consideration that only the cancerous cells are targeted, not the normal cells. Clear? So, with this, we end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.